In this video, we're going to look at how viruses infect us, how our immune system responds, and the effectiveness of vaccines with a more detailed look into COVID-19. Hello, my name is Dr. Freddy Behin, and in the next few minutes, I'm going to take you on a journey into a beautiful world inside of our bodies. I have condensed many years of schooling, including medical school, genetics, biochemistry, immunology, and infectious diseases into this video. But I have super simplified it so you can grasp it at its core. My hope is that with better knowledge and understanding about these pathogens and how they work and how our body responds to them, you will not only stay safe, but you avoid unnecessary panic with all the misinformation that's out there. But before we move any further, I'd like to remind you that this video is purely for educational purposes only and does not constitute medical advice, and as always, you need to consult with your health professionals about any part of this video. Please take a minute to read the medical disclaimer before we move any further. I encourage you to view this video in its entirety all the way to the end, as this is probably the only source you will find with all of this knowledge compacted into an easy to understand video. By the end of this video, you will understand the pathogenesis of COVID-19, mode of infection and how it enters and multiplies in our body, a review of genetics into cellular machinery, whatever is related to transcription and translation of these genes, signs, symptoms, and complications once you're infected, the current available treatment options, an overview of vaccines and how they actually will work, and the challenge involved with COVID-19 virus, and finally understanding the reproduction pattern and the curve of this disease. This video is based on what we know as of May 18th, 2020, and things may change as we gain more understanding about the science of these topics. There is really just one universal common concept in nature, and that is the common goal for survival of every species. That shows itself by certain pre-programmed automated functions, and most importantly, continuous change in genetic information and the structure of proteins within those organisms. This capability of organisms to adapt to their new environment by removing weaker genetic form of themselves and replacing them with newer, stronger ones has allowed some organisms to survive millions of years. So in other words, we as humans are under attack by nature all the time. And if we do not adapt and mutate as we have throughout thousands of years, we will not survive the next environmental changes that are exposed to us. That includes pandemics such as COVID-19. And the same is true for animals, plants, bacteria, and viruses. Every day, we encounter a huge number of pathogens that enter our body. However, in most cases, not only we don't get sick, but we end up having a relationship with some of these organisms in a symbiotic form, as some of them help our bodies perform certain functions and enhance our lives. An example is the bacteria in our digestive tract, which can provide vitamins, help break down food, and most importantly, keep other harmful bacteria in check. To understand how this plays a role in our body, let's first take a look at pathogens. So what are these pathogens that can actually infect you? There are parasites such as worms, protozoas, fungi, bacteria, and viruses. But for the sake of understanding COVID better, we will stay focused on viruses for now. Viruses are the smallest, and hundreds to thousands of them can fit inside of a bacteria or cell. They're much smaller and simpler in structure and design than a single-cell organism like bacteria. They come in many different forms and contain a small amount of genes in the form of RNA or DNA to help them make copies, which I will explain to you later. Some of them are covered by lipid membranes or proteins. Some examples are rabies, HIV, influenza, bacteriophages, which are really awesome, they look like aliens from spaceships, and of course coronavirus. Not all viruses infect human beings. As a matter of fact, some infect plants, some infect animals, and some infect bacteria. The infectability depends on if they have something to attach themselves to, which we call a receptor, and if they're able to bring that genetic information that they contain inside of that cell, and if they can produce something that could cause a cascade of mayhem within the cell. Contrary to popular belief, viruses are not alive, although you can argue that, and there is a lot of arguments out there, but a living organism, by definition, must do the following: perform some type of metabolic activity, make energy from food and reproduce, respond to stimulus physical or chemical or any kind of electrical stimulus and have a cellular composition, and it must grow. So what is inside of a virus? Viruses can exist enveloped or non-enveloped. They have genetic information like DNA or RNA. They could be single-stranded or double-stranded, and they have protein casing. What they lack is no cell membrane, no organelles, no nucleus, and no cytoskeleton. In other words, they lack all the machinery that allows a cell to duplicate itself and perform specific cell-dependent functions. In order for a virus to survive by reproducing and multiplying, it needs the machinery of a living organism. It must attach to a surface of a cell and somehow inject its genetic material to be copied by the host cell. Coronavirus is called corona due to its crown, which is protein spikes on the surface of it. These spikes are sticky and allow them to stick to certain receptors on the surface of some animals and humans. Once attached, they will get swallowed and move inside by the host cell. Keep in mind that there is a specificity about these receptors and these spikes. That's why some pathogens are deadly to some species and completely harmless to others. Fortunately and unfortunately, based on the theories of Charles Darwin and natural selection, organisms adapt to stronger chance for survival due to mutations that occur in their genetics. And as mentioned earlier, that makes them less susceptible to harm and more equipped to survive by coding for new proteins, receptors, and functionalities. One of these mutations has occurred in COVID-19, which is why it has made its way to humans. Let's discuss real quick how genetic material works. Our cells have all the instructions embedded in them to make literally everything our body is made of and needs to function. This information is housed in the cell's nucleus and only represents itself as a compacted chromosome when it's ready to divide. A chromosome is like a tightly wrapped yarn. If you unravel it, you will see the thread. If you zoom in further, you will see that it's twisted around itself for further tightness. Zoom in even further, we will see that it's wrapped and stabilized by histones, and zoom in even further and you can see that there is two railroad-like structures that are twisted in a helical structure. Let's use an example to make this understandable. Imagine you want to build a replica of a home you like. You want it to be exactly the same. You must have a blueprint that specifies exactly how it's going to be built and what type of material and components to use. The entire house is a collection of chromosomes, and depending on what type of an organism you are, you have a different number of these chromosomes. Just like if you're building a one-bedroom apartment versus a high-rise, the amount of information and blueprints needed would be different. In general, the more complex an organism is, the more genetic information it has that needs to be compacted into a chromosome. For example, a fruit fly has 4 pairs of chromosomes versus a human that has 23 pairs. Each chromosome has instructions on specific building material and how they need to be put together. Let's say you want to build a door for this dream house of yours, and the DNA sequence will dictate which door you will be building. Color, size, even if it opens from the right to left or left to right. The instructions in the DNA sequence will also dictate what type of a door handles or hinges you will have. Those are called the proteins that are decoded from the DNA instructions. So our DNA has sections called genes that have specific sequences and instructions that code for something. The gene can code for proteins, a group of proteins, ribosomes, and other cellular building blocks. The building blocks of DNA, which is a combination of many genes, are called nucleotides, which, when mixed in certain sequence and order, present a specific product. For example, if we need to build a cement wall in our house, the components will be a framework to hold the cement until it dries, then the cement, then the water, and something to mix it in. If we're missing any of these components, that wall will not be built. The sequence is also very important. If you pour the cement and build its container afterwards, then the cement will be all over the floor and it won't form. So the frame must be built first, then the cement must be mixed and within the right amount of time poured into that framework to dry. Imagine you put more water or less water? Would the consistency of that cement change? Absolutely. That's why if any of the sequence of these nucleotides are altered, you will have a different product. The nucleotides are divided into two categories, purines and pyrimidines, and they like to connect to each other. Nucleotides are made of three components: sugars, phosphate groups, and nitrogen bases. This combination presents itself as guanine, adenine, cytosine, thymine, and uracil. They like to pair as follows. Guanine likes to pair with cytosine. Adenine likes to pair with thymine and uracil. What's important to note is that DNA has no uracil. Uracil is only present and found in RNA. What the heck is an RNA? RNA is a single-stranded copy of DNA gene sequence which has the base structure uracil instead of thymine. The only difference between a DNA strand and an RNA strand is the extra hydroxyl group on the RNA sugar, and because of that, the base is uracil instead of the thymine. So to distinguish between what we're looking at, all we have to do is look for two items. Also, the name deoxy means no oxygen. RNA exists in three different forms. mRNA is the messenger RNA that carries the genetic sequence corresponding to decoding a protein. The tRNA, which is the transfer RNA, brings appropriate amino acids to build the proteins that are decoded by this mRNA. rRNA, which is the ribosomal RNA, actually reads the mRNA and helps the tRNA to put together the amino acid peptides. In general, when we're talking about RNA, it is referred to as the messenger RNA. So when mRNA is created from DNA, we're calling that a transcription. When the mRNA is converted by the tRNA and RNA to a protein, we call that a translation. So nucleotides make up DNA. Our DNA makes proteins that are building blocks to cell and their machinery. The cells collectively make up tissues, and these tissues make up organs. And collectively, organs is what makes a living organism like us. So what are mutations? Sometimes one or two changes in the sequencing of these DNAs can cause the end resulting protein to either be missing something or have something additional, or simply fold in a different way. Those changes will cause it to either stop doing what it was meant to do or do excessively more than originally programmed to do, or even completely change its functionality and do something completely different. It is due to these changes that species have evolved and that there are so many diverse versions of us around. In the medical world, these mutations can range anywhere from causing cancer to also making you immune to certain dangers out. So, not all mutations are bad. Now that we discussed how DNAs and genetics play a role in protein synthesis, let's get back to our viruses. Sometimes during copying, duplication, and translation phase of a pathogen, errors, reassortment, or recombination occurs. In some cases, these mutations can make the pathogen incapacitated or even more potent and dangerous. But more importantly, when each mutation occurs, the organism may cross over to a new species and infect them. This is called spillover. However, now the pathogen must find new ways to survive the defense mechanism of the new host. And if it is lucky enough to survive, it will strengthen its copies as it moves along, and that's when it becomes an epidemic and pandemic, because the new host has zero idea how to defend itself against this new pathogen that has just crossed over, which is the case for coronavirus that we're facing right now, COVID-19. According to the American Society of Microbiology, most of these crossovers and spills go extinct. But once in a while, after many mutations and recoding of these genes, it will find a way to survive, and that's when we're in trouble. It's very rare for them to survive. Some examples of these successful spillovers that we know of are the following. In the early 2002, a coronavirus that only infected bats crossed over to civets, and later spilled over to humans. This was called the SARS virus because it caused severe acute respiratory syndrome. In 2012, another type of coronavirus from bats also spilled over to camels, and then to humans, and that was called MERS, which stands for Middle Eastern Respiratory Syndrome. In 2019, another type of coronavirus from bats also spilled over to another animal, and we believe that's a pangolin, and then crossed over to humans. We call that COVID-19. There's suspicions that the current COVID-19 may have come from a lab, but as of today there are no solid evidence that this has actually occurred, although we do have the technology to genetically enhance viruses and organisms. That technology has been around for decades and has been used extensively in gene splicing, editing, deleting, using CRISPR and PCR technology. Needless to say, from the moment the Wuhan virus left the part of the world and spread to Europe and US, it has undergone hundreds of mutations. So that means that the same exact virus does not exist in all patients, and we have different genetic strands. Other types of viruses that have spilled over are HIV, which came from chimpanzees, swine flu, which came partially from pigs and a crossover of a bird virus, and the Spanish flu, which was spilled over from chickens. COVID-19 must enter our body either through respiratory system by inhaling it in, through our mouth, or through our eyes. We call this the T-zone. Here's the thing. You have to directly come in contact by touching something and then somehow touching your mouth, nose, eyes, get the virus inside of you. Once infected, the virus makes its way to the respiratory system. Once there, it will move towards the lower part of the lungs, and specifically to the alveoli. Alveoli is where most of the gas exchange takes place, and oxygen and carbon dioxide get exchanged. The coronavirus attaches itself to the type 2 pneumocytes. There are two types of cells inside of the alveoli, type 1 and type 2 pneumocyte. Type 1 pneumocyte is responsible for gas exchange of O2 and CO2. Type 2 is responsible for secretion of surfactant. I will explain what the importance of these cells are in a minute, but first, let's see how the virus hijacks the machinery of the cell to reproduce. Coronavirus has several spikes, and one of which is a spike called S-spike. These S-spikes are important because they have the affinity to attach themselves to the ACE2 receptors. Guess what? Type 2 pneumocytes have ACE2 receptors. Once this binding occurs, it will get engulfed by the receptor inside of the cell, and then it will release its RNA inside. This RNA is a positive sense single-stranded RNA. Let me explain. It can take the rRNA of the host cell and translate and convert itself into a bunch of specific polyprotein molecules. These proteins make up the components that are required to make the final viral structure. The interesting thing about this positive sense single-stranded RNA is that it can also use the cell's RNA-dependent RNA polymerase to convert itself into more RNAs. Guess what? Now you have the components and the original RNA, and voila! You combine them into hundreds of new viruses until the cell can no longer physically hold them and bursts. And these new viruses are released into the alveoli again and infect other cells. This process continues as the surfactant producing cells slowly burst, which results into two important factors. One, the alveoli will not be able to sustain its structure and will collapse. With enough type 2 pneumocytes damaged, the alveoli will not have the ability to sustain surface tension and will eventually collapse. And two, the damaged and burst cells will release inflammatory components which will cause the macrophages to rush in and start their own process. Let's zoom in into that. Macrophages release cytokines that cause an array of issues from there on. These cytokines referred to as interleukin-1, 6, and tumor necrotic factor alpha. These interleukins will cause contraction of the endothelial cells in the walls of vasculature and will open up space in between them and create permeability, allowing flow of content out of these vascular systems into the outside of the alveoli. These inflammatory interleukins will bring tons of neutrophils to try to destroy the virus. These neutrophils will release reactive oxygen species, and proteases. In addition to destroying the virus, now these neutrophils will also damage the inner lining cells, which are the type 1 and type 2 pneumocytes. Remember, the type 1 was responsible for gas exchange. It was supposed to get O2 in and CO2 out. Now with them being damaged, you're accumulating CO2 inside of your blood and you're not oxygenating your blood. So you're gonna get the chemoreceptors to trigger the sympathetic nervous system to increase your heart and respiratory rate, and the symptoms will show as a tachycardia and a rapid rate of breathing. Destruction of these cells will also cause the content of the cells to leak out as debris into the alveolar space, which causes consolidation and will block even more of a gas exchange. This will show up as a symptom of cough. Now you have low surfactant, alveolar edema, collapse of the alveola, which means low level of gas exchange, no O2 is getting in and no CO2 is getting out. Now we're dealing with no mechanical way of oxygenating our body. This is what causes the difficulty of breathing. We must work harder to get any air to flow into the remaining alveoli, which leads to ARDS, acute respiratory distress syndrome. This is what makes this virus very dangerous and can render any medical help we currently have useless. Due to all respiratory complications, ventilators are used to control the breathing for the patient and also help them during respiratory exhaustion, as they have to work very hard to breathe. Also, ventilators help with the pressure to open up the alveoli. However, there are also complications that can occur with the ventilators as well. One of the main mechanisms of expelling microbes, debris, and mucus from lungs is the physical ability to cough and bring those microbes and dead cells out. In fact, one of the complications of ventilators are exactly that. They prevent you from coughing and keep all the garbage inside. Another issue is that the ventilation can cause the healthy alveoli to overexpand and also cause damage. So the pressure control is very important. Let's take this a little bit further. We know that interleukin-1, interleukin-6, and tumor necrosis factor alpha that is released will travel to the brain as well and affect the hypothalamus. The interleukin-1, 6, and tumor necrosis factor will trigger the hypothalamus to release prostaglandins, which in turn will increase your body temperature. This will show up as the symptom of fever. As if this was not enough, as these massive inflammatory processes get severe and they start leaking into the blood and get circulated into the entire body, remember that the inflammatory interleukins will contract the endothelial cells and cause leakage of intravascular content into the interstitial space. As we lose overall blood volume due to the leakage into the interstitial space, the blood pressure starts dropping and you end up with hypotension. Now there is not enough blood or pressure to serve your organs, and organs will start failing, called MSOF, multi-system organ failure. If the kidneys don't get enough blood flow, the BUN and creatinine will rise since the kidneys cannot dispose of them. If the liver doesn't get enough blood flow, the AST, ALT, and bilirubin will rise since the liver cannot dispose of it. If the heart doesn't get enough blood and perfusion, you will see elevated troponin and CKMB. These are some of the lab works that they check for when patients are in the hospital. It all starts with the acute respiratory distress syndrome, which then turns into systemic inflammatory response syndrome, and then eventually into multi-system organ failure. Let's talk about the clinical findings. Patients will show up with cough, fever, it has to be over 104, shortness of breath, tachycardia, hypotension, and lab works that show organ distress or failure. Diagnosis is by a simple oral or nasal swab to rule out influenza A or B, and a real-time polymerase chain reaction, or a NAT. Blood test is going to show BUN and creatinine if kidney is affected, AST and ALT if the liver is affected, and if the heart is affected, increased and CKMB, and troponin. Markers of inflammatory which is increased is CRP, ESR, interleukin-6, LDH, D-dimers, and ferritin. On the imaging, we will see the following. The chest X-ray will show ground glass opacities, which is interstitial edema. That's liquid in the interstitial space. CT scan will show ground glass opacities as well as consolidation, which is the sign of pneumonia or cellular debris. Plus the crazy paving patterns. That's interstitial and alveolar thickening as well as interstitial edema. Ultrasound will show pleural lining thickening, also increased B lines, as you can see here. The more of these present, the higher rate of mortality in the patients. So what are our treatment options? For fever, the obvious choice is Tylenol. Dehydration, we can do IV fluid, but we got to do that sparingly. Remember, we're already dealing with edema as one of the problems. In order to inhibit the entry of the virus into the cell, we can prescribe chloroquine. Ritonavir is a protease inhibitor which will stop the making of the components of the virus. Remember, the virus makes its own components inside of the cell. Remdesivir inhibits the RNA-dependent RNA polymerase, which makes the copies of the viral positive sense single-stranded RNA. Also, zinc can prohibit RNA polymerase activity as well. So we block the entry, we block the making of its components that are needed to make new viruses, we block the copying of the RNA, we have paralyzed the process of the virus multiplication. We also know that interleukin-6 causes a lot of inflammatory responses, which will cascade into a ton of systemic issues. So the good news is we can also prescribe tocilizumab, which inhibits the release of interleukin-6s. Prognosis and risk factors. So as you know by now, all of these little issues can convert themselves into big issues with this small virus. If we have any underlying issues with organs, the mortality rate goes much higher. Obviously, the highest risk is a cardiovascular disease due to how it affects the rest of the body. When it comes to perfusion, control of heart rate, pressure control, it directly affects other organs. Next is any lung issue, which will have much more severe onset in respiratory distress syndrome that we spoke about. If you're immune suppressed, you're on some type of an immune suppressant drug, or have cancer, your risk goes way higher. So mortality rates increase as you have underlying diseases and your recovery rate diminishes. Let's look at some statistics of corona cases versus fatality. The way we calculate fatality rate is the number of deaths over the number of cases multiplied by 100. You should be aware of the fatality rates, as this has become a very big concern in the society right now. We don't want to be overreactive and be paranoid, but also you don't want to be underreactive and take it for granted, as this is a very serious virus. Let's look at SARS virus. With SARS, we had 8,000 cases, 800 deaths, and that gives us a 10% mortality rate. With MERS, we had 2,500 cases and 850 deaths. That gives us 34% mortality rate. COVID-19, up to now, we have 2,250,000 cases and 153,000 deaths. That gives us only a 6.5% mortality rate. You can check these numbers out on covid19.who.int. You should be aware that despite the numbers of death being really high, the number of infected people is most likely not accurate number, and statistically much higher, since there are a lot of people who are not showing any symptoms despite being exposed to this virus. So since they have no symptom, they won't have any reason to go to the hospital and be tested. Therefore, we are missing that important part of the data. So yes, the number looks scary, but most likely a much lower percentage. For those of you who understand math better, know that the larger the denominator is, the lower the result will be. Therefore, since we have a lot of potential people that are infected that we do not have included in our data, that number that we're showing is probably much higher than we should be. Let's learn a little bit about exponential growth of this pandemic. What has made COVID-19 problematic is that it is following an exponential reproductive ratio and spreadability, which we call R0. We believe that the COVID-19 has an R0 of 2 to 3, which means one person can infect 2 to 3 people. That translates into person 1 infects 3 people, those 3 infect another 3, and those will infect 3 as well. That means in 3 cycles, we've gone from 1 person to 40. 1 plus 3 plus 9 plus 27. On the other hand, the influenza virus has a non-exponential reproductive pattern, an R0 of 1.3. The only way to make this curve go down is by decreasing the contact to infected people or have immunity. COVID-19's R0 is greater than 2, therefore it's an exponential growth. Influenza is greater than 1, so it's way slower, and ideal situation is that we have an R0 of smaller than 1. Let's explain the seriousness of this another way. If I have 1 cell inside of a dish that doubles every day, and it takes, let's say, 30 days for this dish to be full, how many days will it take for the dish to be half full? Well, believe it or not, it would be on the last day, day 29th. What about quarter full? Yep, you're right. It would be on day 28th. You see, the growth is small at the beginning and then it follows an exponential growth. This is why when this occurs, our medical capacity to deal with this disease and patients, including supplies, medical professions, and hospital space, become overwhelmed. The good news is that the growth cannot go on forever for the following reasons. It can infect everyone and it will run out of organisms to infect. So it will plateau, following a sigmoid curve, or better known as the logistic curve. Next, some individuals will develop immunity due to their immune system creating antibodies and will no longer get infected by the pathogen. We may also develop a vaccine, isolate infected individuals from reaching others and slow down the spread. What we are worried about is the number of new cases that will cause the health system to get overwhelmed. The midpoint of this curve corresponds with the tip of the infected new cases. If we lower the exposure on a daily basis, we can flatten the curve. Let me explain to you in a visual way. If there are 4 buckets, a bucket for healthy individuals, a bucket for infected individuals, we call I, a bucket for dead infected individuals, which we call D, and a bucket for recovered infected individuals, which we call R. In order for us to slow down and win the race, we must slow down the infected bucket and increase the recovery bucket. The only way to decrease the infected bucket is not to get infected by removing yourself from the environment and not allowing your ignorance and negligence to pass the virus through. This is why it's so important that you share this video with other people so they understand the mechanism of action here. The second is increasing recovery bucket by making sure you're healthy and have a strong immune system. So if you do get infected, your body has the ability to fight the virus and you develop the immunity to it because your body is designed to survive. And if it has all the right components and machineries intact, it will be able to fight the virus. When we get infected with a new pathogen that our body has never seen, there's only 2 things that can happen. We either get sick and recover, or we get sick and die. When we do recover, we have developed immunity to that pathogen, and our body has figured out how to fight it. This is called immunological memory. Unfortunately, this process takes several days. Our immune system is divided into 2 systems, innate and adaptive. Innate is our primary mode of defense. As mentioned earlier, all organisms have one thing in common, and that is their desire to live and reproduce. With that said, every day our body is exposed to billions of bacteria, fungi, viruses, and an array of pathogens who want to make our body their home. The good news is that our bodies have developed an entire defense mechanism, including factories, armies, intelligence providers, weapons, and tons of messengers and communicators who are ready for a good fight to protect you from dying. Our immune system can do many jobs, including communication, inflammation, it can activate cells, it can kill cells, it can remember them and create antibodies, it can do a bunch of stuff. Our immune system also has an army of cells at its disposal Macrophages, neutrophils, mast cells, monocytes, basophils, eosinophils, dendritic cells, killer cells, helper T cells, memory helper T cells, killer T cells, B cells, memory B cells, a bunch of cells. These cells have multiple jobs. They communicate, they activate, they remember, disable, mark cells, kill them, produce antibodies, and cause inflammations. So when an infection occurs, what happens with all these cells? So when an infection occurs, our first line of attack is through macrophages. They intervene. In most cases, macrophages can swallow the entire pathogen whole and break it down inside of them using the enzymes that are in there. During this process, they also release messenger proteins that cause some edema in the regions of infection. That's why we notice some swelling. These messenger proteins also activate neutrophils about the location and the urgency of this infection. So the neutrophils come to the rescue. Unfortunately, neutrophils also kill healthy cells during this battle. They're so dangerous that they're pre-programmed to commit suicide after a few days, okay? And that's when you usually end up seeing some pus in that area. Next, the dendritic cells activate. They're responsible to collect samples of this pathogen and break it down inside and bring the pieces and components out and present it on their surface. These surface parts are called antigens. These dendritic cells can also make the decision whether this is a viral or a bacterial infection. It will then travel through the lymph nodes to get to the helper and killer T cells that are awaiting further instructions to be activated. These dendritic cells look for the helper T cells that has the receptor that will match the part that is exposed. Once a match is made, the helper T cell is activated and causes a massive chain reaction to duplicate itself over millions. Some of them become memory helper T cells for future infections and secondary exposure. Some of these helper T cells also activate virgin B cells in the lymph nodes. Once activated, they will duplicate also in millions and will also activate B cells, as well as memory B cells. Then these B cells start producing millions of antibodies that will have the ability to bind to the pathogen. They can do so because they match the instructions that was given to them by the helper T cell, which received that information from the dendritic cell, which actually had contact with the pathogen and has the actual matching antigen. There are different types of antibodies that the B cells produce. They're called immunoglobulin. We denote them as IG, and then we give it a letter. IgM is for primary response, and that means that you just got to meet the organism that entered your body, and we just figured out what to do with it, and that's the first antibody that gets produced. Once we've gotten to know what this organism is, and the second exposure that we get, we produce IgG. IgA is usually in mucus and saliva. IgE is activated mainly during allergies, and IgD is a part of a B cell receptor. Now let's talk about vaccines. With vaccines, we take advantage of one of the most important part of the immune system, and that is the ability to develop immunological memory. That means that once you have been exposed to a pathogen and survived, our body knows how to recognize that specific pathogen and also knows how to fight it most efficiently. There are only a handful of ways we can make vaccines. We can either have a live but weakened form of the disease, an active form of that disease, part of the disease, deactivated toxins of that disease, or a less dangerous wannabe looking form. Regardless how the vaccines are made, they're all initiating the creation of the protective antibodies. Remember, when infected, our body has to go through primary exposure. It will either make it by producing enough antibodies to fight the pathogen, or it will not make it and it will have a fatal outcome. Vaccinations give our body a chance to go through this primary exposure without getting sick and activate the immune system to create the antibodies needed for when we do actually get exposed to this pathogen. But at this point, our body will consider it a secondary exposure and already knows how to find it and recognize it. We call that the rapid memory immune response. In most cases, vaccines are the safest way to build immunity in a controlled setting. I know in the recent years there have been controversies and debates about the safety of vaccines. Although to date, most credible scientific evidence strongly supports the usage of vaccines to avoid serious diseases in individuals and in population, but if you're for it or against it, I will leave that up to you. That would be for another discussion another time. So why is it a big challenge to create a vaccine for COVID-19? Well, first challenge is that the viruses like coronavirus undergo genetic mutation and as a result change certain aspects of their structure, which is necessary for the vaccines and antibodies to recognize. Next challenge is that the vaccine itself can pose its own unique dangers by activating other unwanted immune responses. As a matter of fact, some vaccines cause a dangerous phenomenon known as antibody-dependent enhancement, which paradoxically leaves the body more vulnerable to severe illnesses after incubation. Some vaccines generate very low levels of antibodies and still leave the host vulnerable. Another issue is that specifically for COVID-19, we need the immune response at the site of infection, which is the mucosal tissue. Not all vaccines promote a strong response from the mucosal immune system. Now that you have discovered and understand more about the immune system, you know that it has evolved over millions of years. It has collected and sequenced billions of structures. Look, in most cases our body has the best ability to heal itself from inside out as long as we do not damage our body with toxins. Have you considered the awful diet we've had in the past 30 to 50 years? Maybe it is time to reconsider a cleaner diet. More raw, more fresh food, less processed and less sugar. Let our body heal from the toxins by being more conscious about what actually goes inside of our body. Many of our health issues, including emotional, neurological issues, start with toxins in our dietary intake with a very low nutritional factors. Let's start thinking of food as fuel for energy rather than something to fill up on. If you are obese and have not paid attention to your weight, I am begging you, it's never too late. Your body is suffering and your organs are going to slowly fail and cause a cascade of health issues. You do not have to starve to get your weight back under control. You can have your cake and eat it too, only if you understand the timing and amount of food intake. It is easier than you think. But you have to stop believing all these fad diets and all these gurus with their programs that are just trying to make money off of you. It starts with a simple decision. It's in the moment of decision that your destiny is shaped. If you're a smoker, I'm sorry, I don't know what to say to you. Not only you're wasting money, but you're also damaging your body and the body of your loved ones around you. If you think it calms you down, I promise, with the right personal development and mindset, you will have no need to cling on to something like cigarettes. If you're leading a stressful life, you're pumping your body with harmful stress hormones that will damage things as time progresses. It's like a ticking time bomb and can cause severe health issues. It is one of those silent killers. Look, I understand very well that dealing with stress is never easy, but it is a part of life. Stress occurs when you believe that there's no options, that you're helpless. So please get some help. There are so many solutions and techniques out there that can put your mind at ease. You might think that you have a valid excuse, but I promise you, the moment you make a conscious choice that enough is enough, life will shift for you. If not now, then when? If not for yourself, then whom? You being the best version and healthiest version for yourself will be a gift to your family and people around you. If not for yourself, do it for the world. Again, I'm here if you need any assistance in any parts. I'll be more than happy to help you in discovering options to get your life and your health back on track. If you watched this far, I really appreciate it, and it would mean a great deal to me if you hit the like button and also subscribe to this channel. I would love to produce more educational videos for you. Please also share this video with your loved ones and get them informed. Again, my name is Dr. Freddy Behin, and it's been my privilege. Live such an amazing life, void of excuses and mediocrity, so that you can be a superhero to someone out there. Because when you win, the world will win. See you next time. What has made COVID-19 problematic is that it's following an exponential these new viruses into and releases the alveoli. Will release ROS, reactive oxygen species. I have condensed many years of schooling, including medical school, genetics, biochemistry, immunology, and infectious diseases into this video. But I have simplified this video so you can grasp it better.